In this video, I'm going to be talking about cadmium. It's actually a heavy metal, and unfortunately, it's in foods like chocolate. So a lot of your grocery store chocolates, whatever you find, whatever brand it is, you're probably going to find cadmium in that. But yeah, I'm probably going to make a lot of videos on cadmium. But a good way to get you should probably get your chocolate tested for any levels of cadmium. But basically, it can be bad for your mitochondria because cadmium can actually it actually has an effect on a PGC1A, which is actually the master regulator of a mitochondrial biogenesis and oxidative metabolism. So immune cells, especially cytotoxic T cells and NK cells, rely on a functional mitochondria for energy so that they can proliferate and kill target cells. Now, energy in the form of ATP. So basically, cadmium is a heavy metal toxin, downregulate expression in multiple cell types, and its mechanisms include oxidative stress, disruption of transcription factors like NRF1, NRF2, which are really good for you, and interference with signaling pathways that normally activate PGC1A like SIRT1 and AMPK. So the consequences for immune cells is that the cell count goes down, which leads to lymphopenia, reduced T and NK cell counts due to apoptosis triggered by mitochondrial dysfunction. Cell count goes down because lower ATP, which is less energy for proliferation, migration, and cytotoxic activity. So the oxidative stress caused by DNA, caused by cadmium, leads to DNA damage, which means impaired transcription of cytokines and cytotoxic molecules. Now, the transcription happens in the chromatin. Inside of the chromatin is where the DNA is at. Of reduced PGC1A, there is an impaired mitochondrial function, which leads to insufficient energy for synthesis and secretion of Granzyme B and Perforin. Basically, what that is that those are like the weapons that immune cells need. Basically, there's the pore, and then the pore is basically what the T cell uses to insert Granzyme B. So Granzyme B goes in through the pore into the pathogen or the cancer cell. As a result of that induces apoptosis. So cadmium stops that from happening. As a result, this reduces the immune system's ability to kill virus-infected cells or tumor cells. Chronic exposure and systemic toxicity of cadmium leads to weakened immunity and higher infection and tumor susceptibility. The bad thing about cadmium is that it also weakens the immune transporters. Immune cells need those transporters so that the amino acids can work, because immune cells also need amino acids to function, like the 9 essential amino acids. So the effect on T cell amino acid transporters is that it strongly disrupts immune cell metabolism, signaling, and visibility. T cells are especially sensitive because their activation requires high flux amino acid transport for mTORC1, proliferation, and cytotoxicity. So cadmium raises oxidative stress, which suppresses SLC7A11. XCT imports cysteine, the precursor for glutathione. So if cysteine can't be imported, then the precursor for glutathione is not going to work. That also means that glutathione won't be able to bind heavy metals. It won't be able to reduce stress. Glutathione has a lot of benefits. So cadmium depletes glutathione and increases reactive oxygen species, which leads to XCT in T cells. The effect is the downregulation of XCT expression because its promoter responds to NRF2 and oxidative stress, but cadmium disrupts NRF2 signaling, which is maybe why you should fast more, because fasting boosts NRF2. So as a result of less cysteine uptake and lower glutathione, the T cells become oxidant damaged. That they may also enter apoptosis. Poorly activated T cells are undergoing stress. The functional outcome is less antioxidant defense, weak proliferation, and lower survival under activation. Cadmium impairs LAT1, which is a leucine transporter. Leucine is an amino acid as well. Leucine is essential for mTORC1 activation, T cell clonal expansion, and cytotoxic Granzyme Perforin production. Like I said earlier in the video, the Granzyme B goes in through the Perforin. So it's like a weapon that the immune cell uses to attack pathogens and cancer cells, leading to apoptosis. The cadmium effect is that it reduces LAT1 transcription via inhibition of ATF4 and CMYC dependent amino acid response pathways. This disrupts membrane integrity, impairing transporter trafficking to the surface, which leads to lower leucine uptake, which means weakened T cell growth and effector function. Also leads to a dysfunction in protein synthesis, which also means that you can't build muscle. Without protein synthesis, muscle can't be built. That can also mean lead to a muscle wasting, or like less muscle, or muscle atrophy. Now that's actually bad because that means that there's less muscle, that there's less muscle mass for a person to be able to survive cancer, because people with more bulk, more muscle mass, have a higher, a better chance of surviving a cancer. Because if someone is so thin and they lack muscle, then that means that they're probably wouldn't survive a cancer metastasis of a lack of a protein synthesis. Also suppresses ASCT2, which is a glutamine transporter, because T cells need a glutamine for energy.